Okay, so I think I skipped this slide. We're talking about vaccines and how vaccines are supposed to mimic the primary exposure to the pathogen, so that when the vaccinated individual sees the pathogen for the first time, that person can respond with a secondary response. We got that far, and then I think I skipped this slide, but basically we're talking about types of vaccines. So one can use whole cells or whole viruses that are killed in some way to make them harmless. And we did touch on Bordetella pertussis, whole cells. One of the two polio vaccines, called the Salk vaccine, is a whole virus that has been killed with formaldehyde. And one of the flu vaccines, one of the two flu vaccines, the injected one is a killed flu virus. You can also imagine using attenuated live bacteria or live viruses. So there's a vaccine against Mycobacterium tuberculosis called BCG. It is a species of Mycobacterium that is attenuated. It was to make it harmless, and it is used a lot. Not so much in the U.S., but elsewhere it is. The other polio vaccine is it's a live polio virus that is genetically weakened to not be harmful. We'll come back to to that question. Also, the other of the two flu vaccines is a live vaccine. You can also imagine viruses that are naturally occurring viruses, but they're not harmful to us. And maybe one example of that would be the vaccinia virus, which is related to smallpox virus. And so people that get infected with it make antibodies that can react against smallpox virus proteins, because the two viruses are similar enough. Subunit vaccines. It's more and more common to find vaccines that consist of a single protein. I think I'm jumping ahead now. I jumped ahead before. Toxoid is basically a toxin that has been inactivated to make it harmless in one way or another. Another thing that you can imagine is to use. We talked a lot about toxins having an A subunit and a B subunit. Imagine injecting somebody with the B subunit. Person with the B subunit is harmless because it doesn't have a warhead, but if you make antibodies against it, that they will protect a person against the whole toxin by coating the B portion of the toxin. So that's possible. Let's go on. Also, adhesins. You can imagine that there are all these. We've talked about things like pili that are used by bacteria to adhere to host cell surfaces. They make good vaccines too, because if the person makes antibodies against them, they will not function to cause adhesion. Okay, so and we talked about the DPT vaccine, and of the three components to it, the middle one, the pertussis, where's my pointer? The pertussis was kind of problematic, and it caused problems. And once in a while, you get children that they just reacted terribly to it, seizures and convulsions, enough so that they sometimes stopped using it. And then what the response was, modern medicine has come up with what is now known as the acellular pertussis vaccine. So what's done, the acellular means no cells. They removed the whole cell. They removed that type of vaccine, and what they have now is this. They have pertussis toxin, which is kind of like cholera. There's a picture. The next slide is a picture of it. It's kind of like cholera toxin, but the point is it's a toxin, and they make it harmless. So I'll come back to that. And they also added in some adhesins. So there's an adhesin it's called. It doesn't matter what it's called, but it's an adhesin. And pili they're also used by this bug. So they add some of those in. So you have this one toxin made harmless, and then you have these two adhesins, and that replaces the whole cells. Now, toxoids. Somewhere I had what a toxoid is. Toxoid is it's a pure toxin and it's activated using formaldehyde. Okay, so the problem with doing that, I think we said before, is that if you have too little formaldehyde is not good, because there could be some active toxin. maybe you don't inactivate it completely. Too much formaldehyde, and you're really changing the shape of the surface of the antigen, and so that's not, the antibodies you make against it are not going to recognize the native toxin. So that's not ideal either. So what has been done is to make it's right there, right? A genetic toxoid. A genetic toxoid is a toxin that has been modified genetically to become harmless. So it's here, I think, right? No. This pertussis toxin. It's a pentamer of B subunits and a single A subunit. And we're not going to go into what it does, but you can see, if you look at that, I'm not going to mention it, but there it is. It's a lot like cholera toxin. Anyway, the point is that the A subunit, 2 mutations are used. So you can change a particular arginine to lysine and a particular glutamate to glycine, and that toxin is catalytically dead. But the shape is unchanged. So that's a harmless toxoid. You don't need formaldehyde, and it's safer and more effective. And the same has been done with other toxins. Okay, now this is a picture of the bacterium, Bordetella pertussis, and you can pick out Filamentous hemagglutinin. FHA is kind of like a pilus, isn't it? And these are fimbri, which is another word for pilus. And so those are adhesins that are used in the vaccine. And there's pertussis toxin. That's the other part. Okay, let's go on. Polio. Poliovirus. We talked about poliovirus, and we talked about how it causes polio, and we talked about how the polio disease is a rare consequence of infection, and it does the poliovirus no benefit whatsoever. The poliovirus wants to minimize the cost to its host and wants to disseminate, wants you to make more of it, and doesn't really benefit from causing any harm. Okay, so it is also a disease of development. What does that mean? It means, look at the graph here. This is cases of polio as a function of time. It was sort of unknown until about 1908. Suddenly the numbers skyrocketed, and then you can look up here, 29,000 cases. It became a real problem early in the 20th century. And it had to do a little bit with, it's a strange story that apparently our water was too clean. We spent all this time trying to clean our water supply and eliminate things like typhoid fever and cholera, and we succeeded in that. But we also eliminate poliovirus, and that paradoxically turns out to be unfortunate, because apparently when infants are infected, they never get paralytic polio. They just have an ordinary brief episode, maybe a little diarrhea, and they're all better, and they have lifelong antibodies, lifelong protection against it, without ever having any risk of polio. That may be partly because of maternal antibodies that they're born with from their mother. And so, yeah, but then of course the water supply gets cleaned up and the polio is removed. And so kids would get it later in life, and at age 10 or 12 or 15 or 20 or 30, you don't have those protections. For some reason, the odds that it's going to get into your central nervous system and cause paralysis are much greater than they are if you get it when you're little. So that's odd. But anyway, so it was a big problem beginning in the 20th century, and huge problem. And of course, President Roosevelt was a victim and could not walk. He was wheelchair bound, and so he declared war on polio. In 1955, Jonas Salk introduced a vaccine, and we said before, it is a wild-type virus that has been killed with formaldehyde, and that is called inactivated IPV. Now wait a minute, I might be injected, because it is injected. I'm not sure what I stands for. It's either inactivated or injected polio vaccine. But the point is, it's a wild vaccine that is killed with formaldehyde, and it gives protection. But about 5 years later, somebody else came out with that oral polio vaccine, and it is this. It is it's a live virus that is genetically weakened, and here's a picture here, right, of oral polio vaccine. You take it, and I can remember as a kid getting this vaccine. They put a drop of it on a sugar cube, and you just eat the sugar cube, and what could be more pleasant than that? So, and it works better. I guess having this oral, this living virus in your system is just it mimics the wild type virus particle more accurately, and you have a better immune response, a stronger immune response, and you have more protection. It does work better. So with these two vaccines, but in particular the oral vaccine, we are almost at a worldwide eradication. So as of 2012, there were only 3 countries in the world that have endemic polio, and those are paradoxically Afghanistan and Pakistan. I mean, paradoxically, you think if the U.S. is going to occupy Afghanistan, at least we can do is cure polio. But okay, whatever. And the third one is Nigeria. For many years, it was a problem in India, but that was eliminated at some point, I guess around 2010. So then in the past year, there's been a little backsliding. So now we have a few cases in Syria, and I think there's some other ones. Yeah, a few cases in other countries. So there's been a little backsliding. Yes, so I guess in 2013 we have these 3 countries, and in 2014 a few more countries. The numbers are still quite small, but there seems to be losing ground. But I guess if you could imagine that the day will come when there is a worldwide eradication. Okay, so this oral polio vaccine is interesting in that it is not entirely safe. So on the right-hand side here, this graph indicates the year on the x-axis and the number of paralytic polio cases. And the dots are total numbers of cases, okay, per each of these years down to 0 in the U.S. And the bars, these gray bars here, are cases of polio, paralytic polio, caused by the vaccine. These are vaccine-associated paralytic polio. These, in 1962, there were 24 cases of paralytic polio that were caused by the vaccine. And there were, in the same year, many cases caused by wild-type virus. Okay, so, but look at after 1980, every case of paralytic polio in the U.S. was caused by the vaccine. Now the numbers are very small, right? 10 cases a year. And it was considered for some years to be an acceptable risk. But then at some point they say, well, look, we don't have polio anymore. Let's stop using this oral vaccine because it does cause cases of polio. How does it do that? Well, I didn't draw this very well. This is this line here is a genetic map of poliovirus. If you think back to the few slides we had on poliovirus, the VP4 and VP2 and 3 and 1 and yada yada, these are all genes in polio. And this is a mutation in the poliovirus that makes it harmless. Okay? So this from it was an A, and it was the Sabin virus has a G at that position, and it makes it harmless, but it can mutate back. So if you get victims of vaccine-associated poliovirus, okay, people that took the vaccine and developed polio, and you isolate virus from those people, they have a mutation from G back to A. That's the wild. A is the wild type residue. So it mutates back in all these cases here, that and that. I guess some of them have other mutations, but the one that really matters is this one. The G residue mutating back to an A gives you back a pretty much wild type dangerous virus. And all 3 of these examples that include that mutation. So the harmless, presumably pretty much harmless, otherwise harmless, Sabin vaccine can mutate back to wild type and can cause polio. And I guess the risk is considered to be tolerable in much of the world. Most of the rest of the world uses this vaccine because it works so much better. And as I say, with that vaccine, we almost have an eradication in the entire world. But there is this risk. So, interesting, I think. What about smallpox virus? Smallpox. We haven't talked about smallpox, but you know, it's a big virus, as viruses go. Hugely dangerous. This is fantastic history. There was a time when people were variolated. What does that mean? What that means is that people would deliberately have themselves inoculated with wild-type smallpox virus by a scratch in the skin. And apparently, if you get scratched in the skin with wild-type smallpox virus called variola virus, your odds of coming through it are pretty good. 1% mortality, as opposed to 20 to 35% mortality if you get it the ordinary by inhalation. So this was considered to be an acceptable risk, and many people would. It was widely done. People would, whole time. Yes, and then that was replaced by Edward Jenner, who used cowpox virus, which is now sort of like vaccinia virus, which is a virus of cows, and he would inoculate people with that, and that would protect against smallpox virus. And the word vaccination comes from vaccinia virus, which comes from, I guess, vacca, which is cow in Latin. Anyway, that's the beginning of the Edward Jenner is the father of vaccinations, and it's widely used. Okay, a little side bit here. Ezra Cornell had smallpox. He was traveling around the Midwest, and he had smallpox. Smallpox had to do the business. But he appeared in Michigan City with a face so marked that I feared it would lead to my arrest. Okay. Also, this is, I think, kind of an interesting piece of history. The evidence seems to be pretty good that Abraham Lincoln was suffering from smallpox when he gave the Gettysburg Address. And you can read this and see if you agree. See if you think the evidence is compelling. Okay. Let me forget this. Well, one thing we can say about this is that we said before that the oral polio virus is not without risk. Turns out the same thing is true of vaccinia virus. You get vaccinated with vaccinia virus, if there's some risk that you won't tolerate it that well. It's a living virus. What does it say? 1/3 of people that get vaccinated feel sick enough to miss work. 1 out of 1,000 get a rash. 1 out of 30 out of 1,000,000 they react poorly. So it's not a risk-free vaccine, and especially the older people are less likely to tolerate this. So there was a theory that in 2003 in the Iraq War that maybe terrorists would get hold of smallpox and we should have the entire country vaccinated. And but the problem was that out of 300 million Americans, that there would be the vaccine would cause a fair number of deaths. So there you go. Other vaccines. Okay, I think we've covered subunit vaccines. Edible vaccines. You can almost imagine potatoes that would express the cholera. This has been done. Potatoes have been made where the potato itself has a large amount of the B subunit of the cholera toxin. Of course, it's harmless because it's the B subunit, and then you can imagine if you eat those potatoes, you might make antibodies against that protein. And apparently, there's something to it. Yes, sometimes bananas too have been made transgenic with. But same thing, in fact, the cholera toxin B subunit has been made to be expressed in bananas, and if you eat them, possibly you get some protection against cholera toxin. This one, DNA vaccines, are kind of an interesting idea. That's this. Look at this. This is a little simplistic here, but the idea is you have a pathogen and it has DNA, and what you can do is take out a gene from it, put it in a plasmid, and so now you have DNA, okay? And you can inject that DNA into a person. And in the cartoon, of course, here we have the blue vector sequence and the brown DNA from the pathogen. And there they are, these little tiny circles. And some of them get inside of the nucleus of the cells of the human, and they make the protein. So basically, instead of injecting a person with an antigen, you inject the person with DNA and let the person make the antigen. And that is considered to be kind of a good mimic of a viral infection. So this apparently has great promise, injecting people with DNA and letting the person make the antigen. It can be done with a hypodermic syringe. There's also these powderless hypodermics. There's no needle involved, and it looks kind of ghastly, but you have these microprojectiles that penetrate through the dead layer of skin cells into the dermis, and the same effect you get. You can actually administer a lot of things with these needleless syringes, and I don't think it hurts as much as it looks. But in this example, we're using that to introduce DNA and letting the person make the antigens, and that's considered to be a nice mimic of viral infection. So there you go, and I think this is yes. So with all these vaccines. This is the schedule of vaccinations of a typical child in the United States, and from birth to 6 years old. And the person at birth is going to get a vaccine against hepatitis, another one, and then in 2 months they get diphtheria, tetanus, and acellular pertussis, and some other ones, and some other ones, and some other ones. And if you add these all up, the number of vaccines, yearly vaccinations against influenza, you get about 3 dozen vaccinations. And there are people that think enough is enough. I'm not going to express an opinion, but I suppose there's some level of vaccinations that might be a bit excessive. If not this number, then some other number. Anyway, the point of all this is, yes, there are websites out there that, you've probably heard about. People are railing against vaccines these days, and a lot of that's pretty much nonsense. But anyway, and one of the reasons that people might oppose some of these vaccines is that we're vaccinating against things that no longer exist. And the reason is that the vaccines have been too successful. They have eradicated from much of the United States, at least, most of these diseases. So we're vaccinating against things where there's no risk. Like IPV, of course, is the injected or inactivated polio vaccine. We have no polio in this country, so why are we vaccinating? Well, there's one reason. And so there's people that are kind of opposing all this vaccination, and they're probably going to be protected because of this. And this is called, I love this idea, herd immunity. If you get vaccinated, the fact that you won't get disease can protect your neighbors. And so the cartoon is kind of nice here. Here we have a population of people that are, they're the blue ones. It says here, not immunized but still healthy, and we have but they're not protected against this disease. And you get, in the cartoon, you get 2 people that are infected and infectious, and within some time the disease will spread, and everyone gets it. But here in the bottom half, if most people are immune, and you get 2 people that are infectious, they're not going to transmit the disease to the immune people because the immune people are immune. And so the spread is going to be limited. So in the cartoon, it's kind of nice that in the cartoon version, okay, this blue person here is too close to this one, and yes, it may spread to that person. But there's 3 other blue people here, and they are protected because the immune people break the spread, the cycle. The spread of the disease is broken by having most of the people immune. So the few blue people, of these 4 blue people here, 3 of them aren't going to get sick because they're protected by their neighbors' antibodies. Now this could be for an artificial vaccination. It also could be in a natural situation. Say it's a herd of deer, and if most of the herd is immune to something, the few individuals that are not immune are protected against whatever. So herd immunity is a big concept in vaccination, and it's also a huge concept in epidemiology against natural pathogens. And okay, I think that's it, because this next one is host epidemics, and that is the topic of the other PowerPoint that is uploaded. So I'll sign this off. Goodbye.